Good evening, everyone. Welcome you all to the week 9 NPTEL live session, which is on doses compensation. Today, also, we are going to do some questions on doses compensation and also some of the questions on long non coding RNA, which we were discussing in last class. So, I hope that my screen is visible to everyone. If my screen is not visible or if I am not audible to you people, then you can let me know by writing in the chat box. Okay, so I think people are joining, so let's wait for extra 2 minutes, then we'll start. Okay, just 2 minutes, after that we can start. Am I audible to you people? Can anyone confirm? Am I audible? Can you hear me? Yeah, anyone can confirm. Am I audible to you? Okay, okay, thank you so much. Okay, let's start then. Let me know if you have any doubts regarding any particular topic we are discussing or anything else if you have problem with. Okay, so first let us discuss about doses compensation. What is it? Because we are going to do a lot of questions on about this, right? X inactivation, doses compensation, and everything. So before that, just a brief description about doses compensation. I believe that every one of you have gone through the lecture videos of week 9, so it will be a brief description. Okay, so what is the meaning of doses compensation? As the name suggests, when there are two copies of a particular gene, so that in a male and female, for example, here if we take your human organism, human, in case of human or mus musculus or anything, here we are observing that both XX, two copies of X chromosomes are present in case of your human female, while one copy of X chromosome is present along with one Y chromosome in case of human male. Now you can see here that human female, if in normal regulation I am talking about, if everything is normal, then we are getting, the human female gets the twice as much products of X chromosome, and this human male is getting only the one particular product of X chromosome. Understood? So here X chromosome is highly expressed, and the, for example, your gene A is present on this X chromosome, then there will be two copies of gene A two twice, but here this gene A will be present in a single copy. So we don't need that. In that case, what happens, we need to suppress one X chromosome to equalize the genetic products between two biological sexes, that is male and female. Okay, so equalize the products of chromosome, that is sex chromosomes, in between male and female. Here in the regulation process, one X chromosome gets suppressed. Okay, random inactivation of one X chromosome. In case of females, one X chromosome gets suppressed or inactivated. So that is called your dosage compensation. Here we are getting the X chromosome products are equal in case of male and female. But if we take this example and just remove this point and take this and this, then we are getting twice as much product in females and in a single product in case of males, which we don't require. Okay. Next, take an example of your Drosophila melanogaster, that is a flies. This is generally used for your scientific studies as a model organism. Okay, here what happens, if you see here, here also this XY and XS is present, but this particular organism, in that case, twofold transcription of single X chromosome happens. So this, as you know that two X chromosomes are present in female, so this fly, what they do, they just increase the expression of your X chromosome in case of male. Okay, twofold transcription of a single X chromosome in case of male, that means it is highly expressed, so that it can equalize its products as comparison to the female. Okay, next example is your C. elegans, or just worms. Okay, these are also best model organisms used nowadays for research purpose. So here also, basically here in case of C. elegans, there are hermaphrodites, that is both male and female are present, and only a female worm. Okay, there is no Y chromosome here. If you can see here, that is O means nothing. Okay, there is no Y chromosome, this is only a single X chromosome, and there are two X chromosome present. So here what happens, decrease the transcription of both X chromosome by half. So this is female case. Okay, this is the female body which has two X chromosome. So in that case, to dosage compensation manner, it just decrease the transcription of these two X chromosomes by half, reduces the transcription process. That means if we just half it and half it, then it will be a single X chromosome. It will act like a single X chromosome, which will be similar to the male companion. Understood? So this is basically hermaphrodite, not female. Okay, means both X chromosome, that means it act both as male and female, and it acts both act as male, not female. Understood? So this is the dosage compensation in three different organisms, that is your human, Drosophila melanogaster, and C. elegans. Now I think you have little bit understanding about dosage compensation. So as the name suggests, when a particular organism has two copies of a particular chromosome, then one chromosome gets inactivated or gets highly activated to compensate with the particular chromosome in other sex. Okay, other companion, like male and female we are talking about. So in case of male and female, we need to equalize the genetic products. Okay, I think it is clear to everyone. If you have any doubts, then you can ask me. Okay then. So this is about dosage compensation, but before moving towards dosage compensation, how it happens and what are the different types of RNA molecules been included in this dosage compensation manner, what is random X inactivation, and what is imprinting, everything we are going to discuss about it. So before that we need to connect with our previous session, right? We talked about long non-coding RNA, not specifically non-coding RNA. So here just few questions about non-coding RNA. After that we'll move towards doses compensation. Okay, so here is the first question of today's session. Which RNA molecule rescue the ribosome in nonsense mediated decay pathway? We discussed about RNA decay, that is when there is a nonsense codon, that means there is, if there is no termination codon is there. Okay, so if there is no termination codon is there and this RNA is just this translation is happening continuously, so in that case we need to stop the RNA or stop the ribosome. So how this ribosome gets rescued? Which RNA molecule help in that pathway? So here are your options: mRNA, tRNA, LNC RNA, or tmRNA. Which one is it? Everyone try to answer this question. I hope we have discussed everything about this RNA decay pathway last week. So if you have any doubt, then we can discuss about it. We are going to discuss. So just try to remember and answer this question. Others, please try to answer this question. You can guess it, okay? No problem. Okay, then let's discuss about it. As I see, some of you have said C or D. I got two answers, that is C or D. So, what is it? Let's discuss. Okay. Uh, we have discussed about mRNA, tRNA, LNC RNA. Now, this new term comes here, that is tmRNA. What is tmRNA? Okay. As the name suggests, TM means it is a hybrid molecule basically. Okay, this is a hybrid molecule with half tRNA and half mRNA. Okay, so this can act as half tRNA and half mRNA. We'll talk about its function also. So tmRNA generally helps to rescue ribosomes. So how, what is the meaning and why does it rescue the ribosome? Because when you see there is no termination codon is there in the mRNA, so this translation process continues and continues and continues. In that case, the ribosome gets stuck. Okay, because for the ribosomes to rescue from the mRNA, there need to be a particular stop codon should be there. Okay, before if there is no stop codon, then how can the ribosome rescue? Because the particular enzymes are don't come to that mechanism, and it doesn't help the ribosome to move out. So in that case, this tmRNA comes into picture. This binds to mRNA which lacks the termination codon. So what are the termination codons? UAA, UAG and UZA. So these are the three termination codons. So in addition, tmRNA adds a degradation signal to the nascent protein. Why does it add a degradation signal? Because you know, if a particular protein, uh, particular mRNA lost its termination codon, and there it is adding more and more number of amino acids, then that is a non-functional protein. It may add different diseases to the cell. Okay, so in that case, it just add a degradation signal so that it will degrade in any RNA decay pathway, and different types of degradation pathways we have discussed in a previous session. So this is the figure. Here we can understand everything how this is happening. So basically, here it is your ribosome. Okay, small ribosome, smaller part and bigger part, and this is your 5 days to 3 days terminal of your mRNA. Understood? And this is your E site, P site and acceptor site, or A site we can call it. So here, at first, different types of ERF3 and GTP complex comes here at the acceptor site, along with your different amino acid chains. Like you can see here, in the peptide side, this amino acid chain have been moving out, and here this acceptor arm, this new amino acids are coming in. Okay, so this is a normal process of your uh, translation mechanism. And here also we can see that UAA, that is your stop codon, is already present. That means if your stop codon is there, then this complex will come to this place, it will bind to the stop codon, and your translation termination happens here only. Understood? But if the termination codon by mistake is not reached, the ribosome gets stuck upon the reaching of 3 days end, and mRNA has to be rescued. So what happened? Now here, see here, there is no stop codon here. Okay, there is no UAA, USG or UZA. So this mRNA got stuck and ribosome got stuck. Now no ERF1 or ERF3 GTP is coming here, so that it cannot be rescued. So in that case, what happens? If you see here, that this tRNA part of tmRNA, so this is a one example of tmRNA. Okay, so I told you that tmRNA has two parts, that is tRNA part and mRNA part. So this red color one is acting as the mRNA part. Okay, this red color one acting as a mRNA part, and this blue color one will acting as a tRNA part. So as we know that tRNA, what, how does it work? It just brings the amino acid to the acceptor arm. Understood? So here this black color one, that is the tRNA part, it brings the amino acid, for example your alanine. Okay, this alanine, this is the amino acid. There must be some your codon region for amino acid will be for alanine will be present here. Okay, for example any codon. So after that, this will continue until this, where it will have one stop codon like UAA. So now, until it reaches that stop codon, now this mRNA is got completed, and this ERF1 GTP complex will come here, and it will just reduce, just detach the mRNA and ribosome. But in the process of this detachment, you can see that a lot of amino acids got added in this process. Okay, for example, if your 10 amino acid are present in the previous protein, now it will be like 16 or 17 or anything else. Okay, for example 6 amino acid have been added to this nascent protein. In that case, this is a non-functional protein or malfunction protein. So this malfunction protein or malfunctional peptide chain need to be removed, so that in that case this ribosome is released, and this tmRNA, it just add one signal, degradation signal to, for example, this may be your degradation signal to this nascent protein, so that the cell can detect that this particular protein is going to degrade because it is destructed. Okay, this is non-functional or malfunctioned. Understood? So in the end, ribosome is released and the newly made fusion protein. So this is a fusion protein because this tmRNA has been added some amino acid from its own only. It degraded due to the signal sequence in the C terminus. Because of in the C terminal, it has added some signal sequences. Understood? So this is the whole process of tmRNA rescuing the ribosome and mRNA. Okay, is it clear to everyone? Do you want me to repeat it? Okay then. See, so this is the RNA molecule rescue pathway of the ribosome by tmRNA. This is your answer, dear. Okay, fine then. Let's move to our next question. What is the function of regulatory non-coding RNAs? What is the function it? So first one is it regulates the transcription, it regulates the translation process, or it regulates the protein distribution. Or D option is all. Try to answer this question. Okay, then let's discuss about it. As I see A and D. So, here you are observing that four options are there, like regulate transcription, regulate translation, and all. So, your answer is all. Okay. Here you can see that regulatory non-coding RNS has multiple function that regulates the act, or they can act as a transcriptional regulator, translational regulator, modulators of protein function, and regulators of RNA and protein distribution. So let's just talk about it a little. So what are these non-coding RNAs? Earlier we have discussed about it. Basically we all know about three housekeeping RNAs, that is mRNA, tRNA and rRNA. Okay, messenger RNA, ribosomal RNA and transfer RNA. So in these three RNAs are omnipresent inside every cell. That's why it's called housekeeping RNA, and it is highly essential for these three RNAs to be present to smoothly maintaining the important function of the cell. Okay, like your transcription, translation, which is specifically required for message transfer. Okay, so but now comes the non-coding RNAs. So what are these non-coding RNAs? These specific RNAs, they are specific towards particular cells only. Okay, so this provides specificity to a specific group of cells. Understood? So this long non-coding RNA, we are talking about long non-coding RNA today. So this they are specific towards certain function inside the cell or inside particular organism. That's why they add some specificity towards that organism. They are not omnipresent like the mRNA or tRNA or rRNA. So in that case, these non-coding RNAs, they regulates or help in transcription. Okay, we are going to talk about it, that how this they are helping in transcription. Also translational regulators, we just talked about one particular RNA, that is your tmRNA. How does it helps in the translation process and rescuing the ribosome in when there is no stop codon is there. Also modulators of protein function. Protein function is multiple functions are there, and I will talk about it shortly. Regulators of RNA and protein distribution. So how does it helps in protein distribution? So generally, uh, as we all know that post transcriptional modification happen inside your nucleus. Okay, after the adding of this capping and tailing process, this mRNA, mature mRNA, transferred to cytoplasm, and in the cytoplasm your translation process happens, and there you get your protein. In most of the cases these proteins are transferred to or move outside the cell to act at a particular location. Okay, so because you know there are different types of proteins and they need to act at a particular location, not only inside a particular cell. So in that case they need a signal peptide. Understood? Signal peptide, this is very essential. And this RNA molecule, if there is like I don't have this figure, but I think you understand. Like for example, this is a one RNA molecule is there. Okay, RNA single stranded. This is, I'm talking about non-coding RNA. Okay, they help in this protein distribution. They add the signal peptide here. They add some signal peptide. There will be some ribosomes, or the protein molecule will be here. Okay, so these are a non-coding RNA act as a thread, and in that case your protein and your receptor and signal peptide, all these act as a bead. Okay, like a thread and a bead, you understand, like a garland, and here there are flowers uh, threaded by using the needle. Okay, like that. So in this process, this non-coding RNAs has important function in distributing the proteins to a different particular location where they need to act also. So these are few cases where non-coding RNAs are specifically required. Understood? I hope it's quite little bit brief description. We are going to talk about the transcriptional regulation. Okay, okay, so let's move to next question. So here is your question about dosage compensation, and here onwards we are talking about the process, mechanism, how it happens, everything about dosage compensation. So in dosage compensation of C. elegans, both X chromosomes are expressed, repressed, partially expressed, or not expressed. In the beginning of this class, we just talked about three organisms: human, Drosophila melanogaster, and C. elegans. How dosage compensation work in these three organisms. So now you can just remember, or you can answer this question as you want. Then we are going to discuss about it again. Try to answer this question. Okay, then let's discuss. As I think that I am getting only two answers, that is C and B. So let's talk about it. What is the actual answer? So doses compensation in case of sex chromosomes to equalize the doses of X chromosome gene products, as I already discussed this, to equalize the doses of X chromosome gene products by means of inactivating one of female X chromosomes in mammals. Okay, so what is the main importance of dosage compensation, or main motive of dosage compensation? Is it to equalize the doses of X chromosome gene products, and in human females one chromosome is inactivated in the form of bar body. Okay, in drosophila males it doubles the expression of genes along the X. That means in to compensate the female gene products, female sex chromosome gene products, the male one just double the expression of your X chromosome in drosophila males. Okay, but in C. elegans both X chromosomes are partially repressed, understood, or partially expressed. Now if we just talk about it by looking at the figures, here also we have discussed that in case of C. elegans, if you see these two X chromosomes are present, and this XO it stands for a male organism, and this double X it stands for a hermaphrodite. That means it can act as both male and female, understood. So they just reduce the transcription process by half. That's why the color is faded here. Okay, the transcription is reduced or partially expressed in both the X chromosomes, so that in both so that the combining both X chromosomes gene products, it will be equal to a single X chromosome gene product, understood. So this is the case what is happening inside your C. elegans. Okay, you can remember it because C. elegans is a model organism in most of the cases, or you can say it's a partially expressed or partially repressed. That is more preferable to repressed. Okay, it is partially repressed. I think it is clear. Okay, next very easy question, everyone can understand, answer this. I think bar body is signified by inactive X chromosome, inactive Y chromosome, partially active X chromosome, or partially active Y chromosome. Everyone try to answer this question. I think it is quite basic and conceptual, and everyone can answer. Okay, then let's discuss about it. As I see that all of you have answered A, that is, bar body is signified by inactive X chromosome. Very good. Okay, so this X inactivation is also called lionization. It is named after the scientist who discovered it, that is Elizabeth Lyon, and there need to be a maintenance of this inactive X chromosome, and we are going to talk about it how this is maintained. Because if in some cases this inactivated X chromosome gets activated, then that female or that particular organism is going to show multiple X products. Okay, multiple or high feminine characters, and which is not a good thing. Okay, so as you know that multiple chromosomes when there is present, like trisomy in case of chromosome 21 leads to Down syndrome. So in that case also, if there is multiple X chromosome or multiple products of gene products more than it needed, that will lead to different syndromes and diseases. So, bar body is named after Murray Bar as he discovered it. And inactivated X chromosome, it is highly condensed. Okay, why it is condensed? Because it the transcription ability of this particular chromosome been reduced or repressed. That's why it is highly condensed. Darkly staining structure that is found in most cells of female placental animals or mammals. Okay, that means in human females also. I think in MSc or BSc lab you have done this experiment, that bar body, to watch bar body under the microscope. I think some of you have done this. Okay, so let's talk about it more. So I think everyone is familiar with XIST. So answer this question. XIST is a LNC RNA, siRNA, miRNA or mRNA. What is it? Okay, let me give you a hint. So this is specifically a X linked inactive specific transcript. That means it helps in X inactivation process. Okay, we are talking about doses compensation. So in case of females, of human females or any other organism, when there is a requirement of suppressing a single X chromosome, so this XIST is specifically required. Okay, X linked specific transcript. So what is it? It is a, obviously it is a RNA, but which type of RNA? Okay, as you said that answer is A. Okay, LNC RNA. XIST is a LNC RNA, that is long non-coding RNA. Fine. So next, answer this also. So this XIST long non-coding RNA is associated with which isoform of histone? H2A, H2B, H2A1.2 or H3? Okay, let's discuss that this XIST is associated with histone isoform. Why I am talking about histone isoform? As you have answered, it is C, that is H2A 1.2. So what is histone? This is a basic protein. Okay, basic means it has basic amino acids inside this protein, like lysine and arginine. They are basic amino acids, and this basic histone, and as we know that DNA, that is deoxyribonucleic acid, it has negatively charged, that is because of this phosphate group PO4 minus. Okay, because of this phosphate group it is negatively charged, and histone is basic in nature, or you can call it as alkaline in nature, so it has positively charged and they attract each other, and because of this attraction histone can able to bind this DNA molecule inside a chromosome. That means it helps the DNA chain. As you all know that DNA is a long chain of nucleotides. So this histone molecule or histone protein that help to condense the DNA chain in the chromosome form. Okay, and this histone acetylation, deacetylation, methylation, these are all histone modifications. So this different types of histone modification helps in this chromosome condensation and decondensation process. Okay, specifically during cell division process in the metaphase stage, the histones get the DNA chain to highly condense to form a visible chromosome forms. Okay, they are highly condensed so that they can undergo different types of cell division stages. Okay, so also we are going to talk about the function of histone, how it regulates the XIST LNC RNA. Okay, so XIST, this is associated with histone isoform H2A 1.2. Generally H2A, H2B, H3 and H4, these are four isoforms generally present, and they are help in regulating the DNA chain to condensed chromosome form. Next, this is the question. So this XIST LNC RNA is regulated by tmRNA, miRNA, T6 or non. Which one is it? Okay, very good. So this is T6. Okay, let's talk about all this in a descriptively manner. So silencing of female X chromosome in mammals. The X chromosome silencing is mediated by this XIST, the X-linked specific transcript, a 16,000 nucleotide long non-coding RNA. Okay, this is the LNC RNA or long non-coding RNA. So this XIST non-coding RNA is recruited complex, has one entry site in X chromosome corresponding to XIST gene itself. Okay, and it appears to recruit a specific histone isoforms like H2A 1.2, which maintains the chromosome in inactive state. Now you know why it recruit histone, because histone are the basic protein which can regulate the chromosome or attract or bind to the chromosome in a strongly manner. Okay, so that's why, because of their positive charged amino acid like lysine and arginine, they are basic amino acids, they are positively charged and they can bind to DNA that is negatively charged in a highly specific manner, so which maintains the chromosome in inactive state. That's why they recruit this histone isoform H2A 1.2. Additionally, this XIST RNA containing complexes recruit histone deacetylase and methylase. Deacetylase means they promote the deacetylation process, and methylases which promote the methylation, that is addition of one methyl group. Okay, so this XI activity is regulated by another 40,000 nucleotide long non-coding RNA, that is T6, and it contains antisense sequence of XIST and therefore is able to regulate XIST activity by base pairing to it. That means they are antagonists to each other. This exist and T6, they both are antagonists to each other as the name suggests. That's why they can regulate each other. That means, for example, uh, if two X chromosomes are present, as we are talking about human female, two X chromosomes are present. So if in a single X chromosome this T6 get activated, okay, before activation of XIST LNC RNA, this T6 get activated. Then this XIST RNA cannot get activated in that particular chromosome, so that it will move towards another X chromosome where T6 is not there, and so that it can repress the other X chromosome by different manners like deacetylation, adding different histone groups to maintain the inactive state, or methylases. Okay, these are the enzymes like deacetylase and methylase. They help in maintaining the inactive state of that chromosome for a longer time being, right? And also this histone H2A 1.2 is specific as essential for maintaining the condensed structure in the inactive form. Okay, so these are few points how this XIST is maintaining this particular inactivation process. So how does this happen? So this XIXT expression is followed by irreversible layer. Okay, remember it is an irreversible process of chromatin modification that include loss of histone, H3K9 acetylation, K transfer lysine. Okay, so this is lysine 9 acetylation and H3K4 methylation. You can remember this H3K4 and H3K9 that are associated with active chromatin, and the induction of repressive chromatin modification including H4 hypoacetylation, H3K27 trimethylation, H3K9 hypermethylation, H4K20 monomethylation, as well as monoubiquitinylation. So these are different types of mechanisms which maintains this X chromosome inactivation process by XIST LNC RNA. So as you know that this is a irreversible process, and in this case this chromatin modification includes loss of histone, acetylation and methylation. This is very very important. You can just try to remember it in the process, like acetylation and methylation, they are the significance of your active chromatin, but they, when we need to repress that particular X chromosome, so deacetylation is important. That means removal of this acetylation group. Okay, and for that deacetylase enzyme is required, and followed by different types of methylation, acetylation. Okay, so just no need to remember all the process, but try to remember the important points like deacetylase is required, methylation is required, because when we are doing the opposite of active chromatin, we are just trying to repress the X chromosome. Understood? So this is all about silencing one female X chromosome in case of mammals. You can go for lecture videos to understand it more and more. I am just trying to brief about it because we know we are going to discuss some other questions also. Fine. So try to answer this. So this XIST LNC RNA initiates the silencing process by moving in a trans mode of activation or cis mode? Which one is it? Trans mode of activation or cis mode of activation? Okay, let's see. So this XIST RNA initiates the silencing process by cis mode. Okay, so what is this cis mode and trans mode? When the silencing process or whatever it is, when the RNA molecule or gene activation, inactivation, or interaction, anything happens inside the gene, that is called cis mode. For example, this is your mRNA, sorry, this is your RNA molecule, and this particular region, that is your XIST, for example, okay, it is acting and or it is affecting this particular region of this RNA molecule or repressing this particular region. Okay, that means it is acting inside the RNA molecule only or inside this gene only. Okay, that's why it is called cis mode. Cis mode. But when, if for example, this is a chromosome and here it is a gene, but it is acting on another gene which is on that is, this is chromosome 1 or this is chromosome 2. So gene A is acting on gene B which is present on chromosome 2. So this will be an trans mode of silencing process or trans mode of interaction. Okay, because it is been happening inside two chromosomes, which two genes which is present on different chromosomes. But if we are talking about a single chromosome, if a single chromosome is there and one gene is acting on other gene which is present in that chromosome only, so this will be called cis mode of activation or silencing process, whatever it is. Understood? If we are talking about two chromosomes and two genes, then that will be trans mode. If we are talking about a single chromosome but two genes, that will be cis mode. So in that case, this is the features of inactive X territory. What are different features? So here this XIST RNA we are just talking about. So this description, what is it? Long functional RNA initiates the silencing in cis mode. Okay, remember it. This is the cis mode of inactive process. For that, this PCG proteins like PRC1, PRC2 are essential, and these are different types of modifications. Just we talked about like H3K27, lysine stands for K. Okay, this is the single um, representation of lysine, one letter code of lysine. this lysine trimethylation, ubutylation, or dimethylation, or whatever. We need to just understand each and everything if you want to go to deeper discussion. So histone variants like macro H2A and histone H1 is essential for this, and we just talked about that H2A 1.2 is essential. Other proteins like SAFA, nuclear matrix associated, and BRCA1 is essential for X inactivation. Exclusion of RNA polymerase 2, late replication, exclusion histone H2A BDD. So here exclusion of RNA polymerase 2, why it is essential? Because when there is RNA polymerase 2 and other enzymes are there, so they will induce the transcription of that particular gene present in that chromosome. If we are removing the RNA polymerase and we want to just suppress the transcription, so in that case we need to remove the enzymes, we need to remove the RNA polymerase and reduce the late replication process, induce that late replication process. In that case, this X inactive process starts, and further it maintains throughout the life cycle of that human, okay, human female. Next question is, calico cat is an example of imprinting, random X inactivation, Y inactivation, or X activation. I think you heard about this example of calico cat. So what is it? Okay, as I got one answer only, that is B, very good. This is random X inactivation. Okay, so let's talk about it. So what is this random X inactivation in calico cat? So there are gene for coat color, okay, coat color of cat. It may be brown, white, black, or whatever it is. Has two alleles. Two alleles means two forms of a gene. Okay, one allele comes from. There are two already. Every gene has two pairs. Okay, one comes from the mother, one comes from the father. Okay, one is paternal and another one is maternal. So this, for example, this is chromosome 1 and gene A, that means it will have two copies. So these two are called alleles. Okay, one is maternal copy, another one is paternal copy. So these two alleles XB for orange coat and this small b, that means it is recessive. It is for black coat. As you understand by this capital letter and small letter, capital letter stands for the highly expressed trait, and small b stands for the recessive trait. Okay, and orange allele is dominant over the black. Therefore, the heterozygous female cats with this particular should have orange color, but it's not the case. In case of heterozygous, color patches in random inactivation of X chromosome in different cells of blastula. So what happens? See here. This is the figure. You can observe and understand it from here. So as you already talked about that this orange coat color, okay, this orange coat color is highly dominant over the black coat color. So now in case of male, if you have this XB, that is orange coat color, okay, so that means the coat color will be orange. Fine. Next is if there is a small b. As you know, there is Y chromosome, it has no role in coat color, so this will get expressed as if it is recessive or dominant, doesn't matter, because in case of male only one X chromosome is there. So if it is dominant, then it will be lead to orange. If it is recessive, then it will lead to black. Fine. So this is very simple in case of male cat. But when you come to female cat, if it has both XB, that is your recessive traits are there, that means it will lead to a black color coat color cat. Here also one X chromosome is inactivated. Understood? Because as we have been talking about, for dosage compensation, one X chromosome need to get inactivated to equalize the products of X chromosome between male and female. Fine. So in case of female, when there is dominant coat color, that is orange coat color chromosomes are there, like XB you can see here. So in case of that also, one X chromosome get inactivated, and in that process this will lead to activation of the orange coat color. So the cat will have orange coat color in case of female. But when so these are homozygous condition, I think you understand, these are homozygous condition. But when there is this heterozygous condition, heterozygous means one allele is for the dominant trait, another allele was for this recessive trait. Okay, in that case what happens, this needs to be according to Mendelism. As we know, in case of pea plant, we understand that if there is a capital T and small t, so this will followed by this capital T will dominant over the recessive trait, and the plant will be a tall plant. Okay, according to Mendel. But here we are observing random X inactivation in different cells of blastula. That means in some cells this XB, this dominant trait, dominant X chromosome gets inactivated, so that this recessive one leads to black color production, while in other cells this small recessive trait gets inactivated, and this dominant color will produce the orange color. Okay, so if we talk about the whole cell of this cat of a heterozygous organism, so this, if there are multiple set, some places this orange color trait gets activated, in some places this black color trait gets activated. I am just marking it as X and Y. So because of this X and Y activation of different cells, that will lead to multicolor patches around the coat of this particular cat called calico cat. Here we are observing multi patches, somewhere it is black, somewhere it is orange. Okay, so this is a random X inactivation. So in this process, sometimes the dominant trait get inactivated, or sometimes the recessive trait get inactivated. According to that, this coat color of this particular cat got changed. Understood? So I think it is clear to everyone why calico cat is taken as an example of random X inactivation. Okay, fine. Next is let's talk about this imprinting. What is imprinting? When we are talking about X inactivation, sometimes what happens, there are two types of imprinting, that is maternal imprinting and paternal imprinting. So what is paternal and maternal imprinting? In that, imprinting means we are reducing the transcription process of a particular chromosome. In that case, as we know that we are getting two copies of every gene. Okay, for example, this is gene A. Okay, this gene A will have two copies. One is from the father and one is from the mother. So this will be a paternal copy, this will be a maternal copy. In this case, when in some cases or some animals, this paternal chromosome, paternal gene gets deactivated, or not deactivated, we reduce the expression of that particular gene, and this maternal gene gets highly expressed. Then in that case, the maternal chromosomes will show its effectiveness. Okay, and in case of lion and different types of other animals, we can observe this imprinting process clearly. Okay, we are not going to detail of this whole imprinting process. I'm just trying to give you a description about this imprinting. That here also, in case of imprinting, see, in case of dosage compensation, we are talking about only sex chromosomes, and these sex chromosomes also random inactivation, not particular like maternal will be get inactivated or paternal will get be activated. Okay, in that case, but in case of imprinting, only paternal will get deactivated or maternal will get deactivated. That means we are targeting a particular allele which comes from a mother or father. Okay, this is the differentiation between imprinting and dosage compensation. Fine, so let's move to next question. So here in epiblast cells, X chromosome inactivation is random, specific to paternal chromosome, specific to maternal chromosome, and non. Epiblast cells are those cells, this is the term from developmental biology. So epiblast cells are those cells which will lead to production of the embryo. Okay, which will lead to production of this ectodome, mesodome, and other endodome, and that will lead to further production of your gastrula, and the cell differentiation process will there. Some part will move to produce the heart, some part will move to produce the kidney, like this. So the differentiation process, these are the epiblast cell which will lead to production of the actual organism or embryo. Okay, so in that case, this X chromosome inactivation is random or specific to paternal or maternal X chromosome. Okay, let's discuss about it. Very good, you answered it correct. That is random. Okay, we are just talking about that X inactivation process is random. But when we are talking about imprinting, it can be paternal or it can be maternal. So imprinted and random X inactivation, what is it? So in trophoectodome and primitive endodome of blastocyst, X inactivation is subject to imprinting of paternal X chromosome. In epiblast cells, this X inactivation is random, which will lead to production of the embryo. But this trophoectoderm endoderm, they can be lead to different other accessory organisms, sorry, accessory parts. Early XIST expression is exclusively of paternal origin. The maternal XIST allele is repressed until the morula stage. Fine, no need to go into deeper or get confused between this. So this, understand that this X inactivation for dosage compensation, it is random, but while we are talking about imprinting, so this can be a paternal or maternal depending upon the organism. So this X inactivation starts from XIC, that is X inactivation center, or T6, or XIST, or centromere. Which one is it? Okay, let's discuss about it. Very good. That it is, the answer is A. Okay. So X inactivation, that is X chromosome inactivation center locus on X chromosome, and it contains non-coding RNAs like XIST, T6, and JPX, that is activator of XIST. You can remember it as JPX. Next is FTX, that is the 5 prime to XIST. Okay, so XIST gene transcribes a long non-coding RNA XIST, that is 17 kb in humans. As the name is XIST, also the long LNC RNA name is also XIST. No need to get confused. So this XIST RNA is spliced, polyadenylated, but is not translated. Okay, and it has, this XIC has 12 genes, 7 proteins, 5 untranslated RNAs. So this XIST and T6 are the untranslated RNAs. That means generally what happens, uh, after transcription, every RNA move to post-transcriptional modification, and after this post-transcriptional modification, they will move to the cytoplasm for translation process. But these are untranslated RNAs because they have important role in the inactivation process inside this chromosome only. On the active X, T6 expression antagonize the exist. That means, as I was telling you, that when there are two X chromosomes in human female, for example, if one X chromosome, this T6 get activated before XIST. So T6 regulates the XIST function. That's why it will antagonize XIST, and in that process XIST will not act on that particular chromosome. It will move to another chromosome, X chromosome, and repress it or reduce the transcription process. Counting and choice of X chromosome is very important. Why it is important to counting? When, for example, if a human female has 3 X chromosome, okay, this may happen that it has 3 X chromosome. Then in that case, this XIST RNA need to repress 2 X chromosomes, not one. If we just get repress the one X chromosome, then it is also creating the same problem. Okay, so for that case, and we don't need that, so counting is important. What are the number of X chromosome, and depending on that, for example it has 3, then it need to repress 2 X chromosome. If it has 4 X chromosome, then it has to repress 3 X chromosome and leave one for the activation process. Okay, that's why counting is important, and choice of X chromosome depends upon the T6 and XIST RNA regulation process. This is the last question of today's session. So, stability of this XIST transcript is brought about by promoter, terminator, proteins, or RNA polymerase. So, there need to be maintenance of this XIST transcript, right? So, how they got this stability? Okay, let's see then. So, this stability of XIATS transcript is brought about by the promoter. Okay. So this X chromosome inactivation center, it increase in stability of XIST transcript is brought by developmentally regulated switch in promoter use. So this P1 and P2 are used for transcription of stable XIST transcript, and P0 is used for unstable XIST transcript. Okay, these are the name of promoters, that is P1, P2, these are the name of promoters which is used for stable XIST transcript or unstable XIST transcript. And 2.12p reason, so histone H for hyperacetylation in undifferentiated female uh, endoderm stem cells. Okay, so hyperacetylation disappears upon differentiation, and stabilization of XIST RNA might depend in the levels of XIST RNA present in the cell. Fine. So this is the whole process of X chromosome inactivation. If you see here, okay, uh, lines and signs, they are long interspersed nuclear segments. So this is the XIC, that X chromosome inactivation center, and this inactivation process starts from here. This, for example, this is the X chromosome which we want to suppress or which we want to get inactivated, okay, using the XIST RNA. So here these are the blocking factors present already, and XIST, as we know and we have talked about it, it is responsible for inactivation process. It start the inactivation process, as you can see, and moved like a wildfire. And so this is the cease mode of inactivation, that is, as you can see here, this XIST is acting on each and every gene segments present in the X chromosome. So this is in cease mode of inactivation. Okay, XIST RNA coating in cease mode. Fine. Next, establishment of the inactive state and asynchronous replication. Asynchronous means that is it removes from the synchrony. Every gene and cell is present inside the human body in a synchronous manner. Okay, they help, and the synchronous manner regulates and maintains every process. So the establishment of inactive state asynchronous replication is there. Okay, so now it just reduces the replication process in a and move it to a asynchronous manner. Next, what is it? Macro H2A recruitment, histone H3 and H4 hypoacetylation. So here hypoacetylation happens. So in case of hypoacetylation, this reduces or condenses the size of chromosome and move it like a bar body, or you can call it as a highly condensed chromosome. Now you can see that what is the length of the chromosome in the beginning. Okay, this is the beginning of X inactivation process. You can see here, this is the length of the chromosome. After that the length reduces, and while it move towards hypoacetylation, earlier we talked about that in case of activation process, acetylation and methylation is important of X chromosome or any other chromosome. But here this histone hypoacetylation just condenses the chromosome more and more, so that it cannot move towards replication, sorry, replication and transcription process and all. Understood? So this is the whole process of X chromosome inactivation. Got it? So this is all. Uh, is it clear to everyone? Do you understand the X chromosome inactivation process? Okay, very good. So this is all. Today we have discussed about X chromosome inactivation, dosage compensation, including random X inactivation, some kinds of imprinting also. And earlier we talked about LNC RNA and how this tmRNA helps in rescuing the ribosome. Okay, if you have any doubts regarding this, you can ask me. Okay then, see you in next class on next Monday. Till then,